Sharma, doctoral student in computational biology in Second University of Naples. We will be presenting about the protein-protein interactions of certain proteins. Such proteins are known as silent information regulators. They are discovered in Saccharomyces cerevisiae. They are having 7 Sirtuin homologs. They are intrinsically disordered proteins. They are highly conserved during the course of evolution. So they are independent deacylases. Majorly Sirtuin 1, 2, Sirtuin 6 and 7 are nuclear deacylases, where Sirtuin 3, 4, 5 are mitochondrial proteins performing ADP phosphorylation as well as deacetylation. So they are localized in nucleus, cytoplasm and mitochondria. So recently it has gained a lot of importance as they are extending longevity up to 30% in C. elegans and the yeast, and potentially they can be enhancing our longevity in humans. They are implicated in cancer, neurodegenerative disease, inflammatory. Recently they have known to be involved in diabetes type 2, diabetes mellitus type 2. So we accumulated the data from across various databases like HPRD, BioGrid, MINT and pathway interaction database, which have experimentally proven interactions or the interactions that are accumulated through high throughput methodological technologies. And then there were some interwoven networks inside this Sirt interactome, which was obvious that they are implicated in the cancer, neurodegeneration and aging specifically. We couldn't have the neurodegenerative data set, and there is not any more studies on these interactions yet. And the kinases, acetylation and methylated subnets are retrieved from phosphocytoplasm and the kinosome. So the dataset was visualized by a cytoscape software. The centrality statistics was done by network analyzer and Centiscape. Then the modular organization of the network was done by Modular framework, and then the functionalization of the network was done through gene ontological analysis with time, which means that they were scale-free networks and were small world. They were responsible mainly in the chromatin remodeling or the cytoskeletal reorganization and metabolic processes. But in these studies, I'll focus more on the human Sirt protein-protein interaction. It was having a vast number of interactions, direct and indirect. There was around 5,786 proteins interacting between them in this network in second order. And there was around 243,365 links between these proteins. The circles you can visualize are Sirtuin 1, Sirtuin 2, Sirtuin 6. This is Sirtuin 3, Sirtuin 5, Sirtuin 4 and Sirtuin 7. Then the statistical insights on the networks. This average clustering coefficient, which gives the measure of the degree how it clusters around the network. It was decreasing, which means that most of the proteins was highly clustered, which you can see that these are the number of nodes and this is the average clustering coefficient value, and these are highly clustered nodes. So then we checked for the node degree distribution, whether it followed the noble biological paradigm of being a scale-free network. It followed the power law, which means that there are hub proteins which are interacting with low vertices protein rather than cells. Then there were between centrality, which checks which is the central component of the network analysis. It gives the load placed on a particular node, which gave the red one is TP53, which is a protein 53, which is having the most loaded protein in this network. Then there is EF1A1, which is eukaryotic translational factor 1 alpha 1, which helps in the nuclear export. And then there are kinases. Next, there was average path length, which is very low. In fact, it is 2.6 per protein. If one protein has to interact with the other protein at a peripheral protein, it only takes 2 steps to interact with any of the protein in the network. So, which is very slow. We also calculated the neighborhood assortativity, which gave the assortativeness of the network. which means how a neighbor interconnects with them. So that gives some increasing trend, which means that there might be some hub proteins that are interlinked in between them rather than interlinked with the low degree vertices. So we found that there are certain proteins, but most of them were following the Barber-Shierberg model or the CM3 model. So the proteins that was hub proteins that was interlinked with the hub proteins were eukaryotic translational factor. Then there was some chaperones like HSP9-TAB, HSP5. Then there was again HSP9. Then there was casein kinase 2A1 and calmodulin 1. So this was the subnet that we had of the rich club protein, which was highly interacting with each other. Then we had insights on the interlinkedness of the hub proteins. So we wanted to check about the modular organization of the network, whether these proteins are important in this whole big network of Sirt proteins. We found there are 20 odd modules that was formed. So the highest value was for the EEF1A1, which is the nuclear export signal, which shows that Sirtuins might export from nucleus to cytoplasm to carry out their function. Then this GABRB2, which is a protein responsible for the cytoskeletal or signal transduction, this was showing the highest modular overlap, which means it was the component of this module was implicated in most of the modules present in this, and these lines suggest the overlap between the various modules. So then we carried out the analysis to functionalize these modules, that what are their potential functions. We checked with the consensus. The Sirtuin, they were having a very mixed function, whereas the module 1 was having all the proteins that was interlinked hub proteins, and they were basically showing function, molecular function related to ATP binding or the metabolic pathways or protein binding. Then there was Sirtuin 3, which was histone deacetylase 1, which was showing transcriptional regulator, and then there was this module 8, which was at the center showing oxidoreductase activity. We tried to check whether in which module does these Sirtuins are present. We found out that these were present in the central modules of this modular organization, which was functioning in the metabolic pathways, and then the transferase activity, or then the oxidoreductase activity. Rather, Sirtuin 2, which was less studied than Sirtuin 1, was showing the largest modular overlap among Sirtuin proteins, which potentially shows that Sirtuin 2 is more functional than Sirtuin 1. Then we check for the network skeleton, that how robust is the network skeleton of these proteins. The first network gives the information transfer between the proteins. We took top 50 proteins out of the whole network, which was behaving in information transfer, and then the top 50 proteins which were at the core of the network skeleton, and then the interlinked hub proteins. And we deleted the proteins from other two networks. Like if we have the 50 community centrality proteins, then we deleted the proteins that are present in the betweenness centrality and the interlinked hub node. What we found out was that the information transfer nodes and the core skeleton of the network was unaffected in this analysis, and only interlinked hub proteins was affected, which showed that these proteins are either of high information transfer proteins or either are at the core of the center. So they are highly important proteins in this network. If you mutate, in biological sense, if we have to speak, if we mutate or delete or dysfunction these proteins, this will affect most of the network, and will be helping in the proliferation of the disease conditions or cascading behavior. Then there are subnets in the network which was of cancer aging. We will first focus on the cancer subnet, which was having some proteins that were acetylated, then there was some kinases, there was some methylated proteins, housekeeping. There was some protein that was showing the double functionality, like there were some acetylations, acetylated proteins as well as behaving as a kinases. So this cancer subnet was retrieved from the cancer gene census of Wellcome Sanger Institute, and they were the subnet was comprised of 303 nodes with 1,678 edges in between them. As you can see, that the average path length of this network was very high as compared to other networks, which we will be following later. So it showed that these proteins were sparsely connected, which means the networks are highly robust. They are not at the center. If you perturb any one of the protein, you cannot have a cancer condition. It needs a whole system to be perturbed. Then there was a clustering coefficient of 0.319, which is a very high clustering coefficient. So they are highly clustered in one community. The diameter was 6, which was at the highest that biological networks can have. So the bottom line or take-home message from this network was that this protein related to cancer occupies a peripheral position in the second-order Sirt interactome. Then the centrality statistics followed a novel biological paradigm, having decreasing trend in average clustering coefficient, decreasing trend in degree distribution, which showed that they are scale-free and small world. So one more phenomenon was that there was some hub proteins calculated in this. On the basis of degree, there are EP300, which was directly interacting in with Sirt proteins. Then there was FP, and then TP53. So there were some proteins that were directly interacting in certain interactome, which was showing the bottleneckness in aging, which means they are having a high load in aging subnet. And these proteins, in this subnet, 66% of the proteins were implicated in DNA binding, then 45% of the protein was performing ATP binding, and 40% of the proteins were involved in the transcriptional regulation. Then the post-translational modification subnet, there was kinase subnet. So the global view of the network statistics, we found that there are a large number of average papers in Sirt family interactome. Then the cancer subnet exhibited the highest average path length, signifying the low degree vertices to be localized at the periphery of the Sirt second-order network or any other biological network you can say. Then there was a phenomenon with aging subnet joined that this subnet was quite homogeneous as compared to the other subnet, and it was having a very shortest path length, which showed that this aging subnet can synchronize very well with the perturbations or with the normal functionality of the biological processes. Second-order interactome cover almost 25% of the human proteome till now, when there is only 10 to 20% of the interactions known of the protein. The network is scale-free, has a scale-free degree distribution, which means they are quite robust. Targeted attacks would disrupt the network, while the random attacks will be vulnerable on the network. There was 20 overlapping modules showing diverse functionality, suggesting pleiotropic functionality of Sirt proteins. Then 4 modules demonstrated mixed function, especially ATP binding, cytoskeletal organization, or the transcriptional regulation. 10 core proteins of 4 modules, eukaryotic translation factor 1 alpha 1 comprised of interlinked hub proteins. And disease related to protein, except hub proteins, are at the periphery of the Sirt second network, especially the proteins of cancer subnet, were very sparsely distributed. Thanks, this was the group that I worked for 4 months on the modularization of the network, and this is my host lab in Second University of Naples. Thanks.